Hey everybody, and welcome to chapter 3. This is the first part of chapter 3 where we're going to explore the body cells, and this is broken up into several sections because it's a fairly large chapter. We are studying this though because it's the next level on the hierarchy of organization. We've been through this chemical level so far with atoms to molecules and then macromolecules. And so in this chapter, we see how those macromolecules form organelles that are part of cells. And we are in cytology, the study of cells. And so we can't really start without going through the cell theory, which says that cells are the basic unit of life. And cells are alive, cells are living organisms. And in the human organism, we have trillions of cells, and we have over 200, or as your text says, 250 types of cells. In this chapter, we're just going to look at the general animal cell model and what that sort of looks like. But we'll see more of that complementarity between the structure of cells and their functions. But just as a reminder here from the slide, the continuity of life has a cellular basis, which means that cells come from pre-existing cells. And we'll go through how they are formed in this chapter as well. So cells, again, are very diverse, but in this chapter we just kind of look at a very general animal cell model. But as cells diversify, they become specific for specific body systems, which allows them to carry out specific functions. So on this slide, we see some cells that you might recognize, maybe some that are new, but skeletal muscle cells are very, very different than what we'll see in chapter 3. These are long, tubular shaped cells, and they actually have multiple nuclei in the cell, not just one. Erythrocytes are red blood cells, and they actually eject their nucleus as they mature. Sperm cells have a flagellum, right? And so we see some modifications in the shape and the structure of these cells that are going to give them unique functions. The flagellum helps the sperm to swim. The biconcave disc shape of these red blood cells helps them to be able to squeeze through capillaries really easily. And the long tube-shaped structure of skeletal muscle cells helps them to be arranged in parallel kind of fashion, and the multiple mitochondria inside help them to make ATP pretty efficiently. So a cell generally has three parts. It's got an outer plasma membrane, and then it's got a nucleus inside. We said in the last chapter that's where the genetic material is stored. And then sort of between those areas, there is cytoplasm. So cytoplasm is the fluid part of the cell that contains organelles, specialized organelles that carry out the cellular functions. The plasma membrane creates a barrier between the fluid inside the cell and the fluid outside the cell. And as a heads up, the fluid outside is called extracellular fluid, or ECF. The fluid inside is called ICF. And so this is a characteristic of living things too. We've got to have this barrier that separates the internal and external environments in order to maintain homeostasis. So wouldn't it be nice if that was all you needed to know about cells? It's not. So we've got to go through more organelles, and we'll talk about them kind of individually. In this picture, though, we see the plasma membrane around the cell, the nucleus, and then within the cytoplasm, we see a fluid part called cytosol if we look kind of closely, and then lots of organelles. Okay, so let's talk more about the extracellular material. Where is that? It's kind of neglected because we talk so much about what happens inside of the cell, but outside of the cell, there's important extracellular materials, not just fluids, but also some other components, including the matrix, the extracellular matrix. This will come up in chapter 4 in a big way, because in chapter 4 we talk about when you put two or more cells together of the same type, same functions, they create a tissue, and so tissues have unique matrices. All right, extracellular materials though include extracellular fluid, which includes something called interstitial fluid, and that's the fluid in which the cells are kind of bathed and submersed. Blood plasma is technically also an extracellular fluid, And this is the fluid matrix part of blood. It's about, well, let me go back. It's about 55% of the content of blood, and the makeup of plasma is about 92% water. There's also some proteins in there and some other things. Cerebrospinal fluid is another example of extracellular fluid. This is the fluid that nourishes and provides buoyancy to the brain and spinal cord. So let's talk about the plasma membrane first. This is our barrier that separates ECF and ICF, and it controls what goes into the cell and what leaves the cell, and sometimes it's just simply called the cell membrane. So the makeup is a fluid mosaic sort of bilayer of phospholipids. All right, so let's look at that. And you can sort of see here our little, I know the picture is kind of small, but it's 2 layers of phospholipids. And those are a class of lipids, and that makes up about 3/4 of the structure of the plasma membrane. Now, that's not just it, but we'll talk about this for just a second. These phospholipids have heads that face outward. They're hydrophilic. They can interact with the water well, and that watery ECF and ICF. And then their tails face each other, and they are hydrophobic. So that's a big part of the membrane. Now, these can switch places very, very easily. There's also proteins within the phospholipid bilayer that are embedded. Some of them go all the way through the plasma membrane, and some of them are just on one side. But these act as cell markers, which allow cells to communicate. They act as channels through which things will move into the cell or out of the cell. And so these can switch places. Because of the appearance of the cell membrane and the different macromolecule kind of structure, and the fact that they can move and switch places a crazy amount of times per second, by the way, it's often referred to as a fluid mosaic sort of pattern. Now, there's not just proteins and lipids here, there's also carbohydrates. And that includes this sugary kind of projection from the surface of the cell called the glycocalyx that also helps out with cell-to-cell recognition and communication. And we'll talk about cell junctions in a little bit. Those are mainly the product of proteins. Okay, so again, the phospholipid bilayer is made of about 75% or 3/4 of phospholipids, which have hydrophilic polar heads and fatty acid tails that are nonpolar and hydrophobic. There's also glycolipids in here, and these are basically carbohydrates and lipids sort of combined, and they help out with acting as cell markers as well for cell-to-cell communication. Lipids also include cholesterol here. About 20% of the lipids that are found in the plasma membrane are cholesterol, and this is super important because it stabilizes the phospholipid bilayer. So this picture is much better to show you what those phospholipids look like close up. And so we see a double layer of them all the way around the cell. Of course, it wouldn't be, flat. It would be a sphere-shaped structure, and they would surround the entire cell in this way. And this makeup is super important. It will come back up in later chapters too, especially in the endocrine system when you talk about how some hormones that are hydrophilic can interact with cells and how hydrophobic ones do. All right, so let's go back to proteins for a second and talk about them some more. These allow cellular communication both with the environment and with other cells. They make up about half of the mass of plasma membrane, and they have specialized functions. Some of them are hanging out sort of freely around cells. Some of them are tethered or kind of anchored down to intracellular structures. And so there's 2 types of membrane proteins. Integral proteins, they are all the way through the plasma membrane. So they're embedded and kind of go through both layers of the bilayer. And then peripheral proteins are just on the periphery and extend from one side of the membrane. So these both can function as I'm sorry, let me go back. Sorry, receptors. Can't talk this morning. But they can also act as enzymes. and they can function as transport proteins, but integral proteins, since they go all the way through the membrane, they're better suited for that role. So peripheral proteins are enzymes in some cases. Sometimes they can act as motor proteins, especially during the cell cycle and cell division or skeletal muscle contraction. And they help out with acting as connections that hold cells to other cells as part of cellular junctions. And that's what helps to make tissues too. So these membrane proteins, again, can help out with so much. Membrane transport. They can create channels through which ions and electrolytes move in and out of the cells. And there's animations on mastering and in your e-text for that. They can act as receptors that help out with signal transduction. We will see that in other chapters and even in ANP2 as far as like hormones that bind to these receptors and activate some sort of response in the cell. They can act as enzymes that catalyze and speed up reactions. And they can help out with cell-to-cell recognition. That allows cells to communicate with each other, and they can also act as cell-to-cell sort of anchors that attach different structures to the cell. So in this picture, for example, we see attachment to the cytoskeleton and the extracellular matrix. This will make more sense in chapter 4 when we talk more about the matrix and what it is. So I'm going to sort of hold on to that thought for later, and we'll come back to that in chapter 4. Cells can also be anchored in place together by their plasma membranes thanks to these proteins in the membrane. And this creates about 3 different types of cellular junctions. And so more to come with that too in chapter 4. I think it's in chapter 4. It may be later in chapter 3, but we'll talk about what these junctions look like, how their structure suits their function. But these can be a really tight connection or tight junction, or they could be a little bit more loose. I will say this as a preview to the cellular junction stuff too. Sometimes there are proteins that have just a big channel, and that allows material to move from the ICF of one cell through these gap junctions into the cytosol of another cell to allow communication in that way. Okay. So let's talk a little bit more too about the glycocalyx. This is the glyco tells us it's sugar, and so it's carbohydrates that are essentially protruding from the cell's surface. Some of them are anchored to lipids or fats, and so those are called glycolipids, and some of them are anchored to proteins, and so that's called glycoprotein. But these help to act as markers that help cells to recognize each other and help the cell to interact with their environment. This also is important for the immune system because it helps cells to be recognized as self or non-self. And so your book goes through this scenario of what if something goes wrong with a glycocalyx. So sometimes cancer cells can change so fast that the immune system and the immune cells and proteins don't recognize that glycocalyx of the cell as being damaged. And so then they're able to continue replication without control. Okay, so very important glycocalyx, including the glycolipids and glycoproteins. And here we are at cell junctions. Okay, I thought it was chapter 4, but here it is. So cell junctions just link the cell plasma membranes to each other to hold cells together, and that helps to form tissues and eventually even organs. Now, we see this especially in like epithelial tissues, but we don't see it in some types of cells and tissues like blood cells and sperm cells. But there's 3 types of cell junctions. So we've got tight junctions, desmosomes, and gap junctions. And we'll go through tight junctions first. Let's show a picture of what this kind of looks like. So with tight junctions, it's like it sounds. There are cells that are held together really tightly. These proteins really anchor the plasma membranes of adjacent cells together tightly. All right. And so they're kind of fused together. The plasma membranes are almost fused together, and that keeps the fluids and other materials from being able to move easily in between those cells. And so you find tight junctions in lots of places, including the blood vessels that are in the blood-brain barrier. And so this is useful because it allows or doesn't allow the passage of material between those cells to enter the bloodstream. All right, we also have desmosomes. This is a great description. They're hard to kind of describe sometimes, but your book says a rivet-like cell junction formed when linker proteins of neighboring cells interlock like the teeth of a zipper. So a zipper is a great analogy. Button-like is another one that comes up here. Let's look at the picture from the text. So you can see how these proteins are holding these plasma membranes together. Now, what's the purpose of this structure? Well, it provides a lot of mechanical strength and resistance to abrasive sort of forces. And so we find desmosomes in different areas of the body, but especially in places like the skin, where it reduces that likelihood of tearing that tissue because of stress or twisting or tension. Right, and then the last cellular junction is the gap junction, which I tried to explain a while ago. You've got two cells, and hopefully you can see this image if you're able to. So what's happening in this picture is there's two cells side by side. Of course, their plasma membranes are connected by proteins, but the protein has a channel, and that channel runs from one cell's ICF to the other. And so that allows material to move from the ICF of one cell through the channel into the next cell. And that allows for really easily communication between the cells, spreading impulses. So we find these types of junctions in places like the heart. So it allows those electrical signals to move with the help of ion movement from one cell to the next really, really easily. All right, so we're getting into membrane transport next. We'll go through a little bit of information here and maybe split it up a bit, but many substances have to be constantly going through the plasma membrane. Some of them can go through easily, some of them don't, because the plasma membrane is selectively permeable. It doesn't allow everything to move through, and it can select what it is permeable to. So there's two big, broad ways that things can go through the plasma membrane into or out of the cell. And that's passive transport, where no energy is used by the cell. That includes things like osmosis, simple diffusion. And then there's active transport, where cellular energy has to be used for that transport across the plasma membrane. So let's go through a few examples of passive transport next, and I'll share some animations. Your text has some animations for these too. But passive transport, again, has no energy that's used by the cell. So there's simple diffusion, facilitated diffusion, and osmosis that are categories or examples of passive transport by the cell. So all of these involve diffusion, which is just the movement of molecules from an area where they're more concentrated or more densely kind of populated to an area where they are at lower concentration. And so since they're moving from higher to lower concentration, they're going down a concentration gradient. And a gradient, again, is just the difference in the amount of something between two adjacent areas. Okay, so all molecules have random high-speed movement because of kinetic energy, and so there's collision between these molecules. But if molecules are packed together in higher concentration areas, then it will make them collide more often, and so that helps the molecules to be scattered as they move into lower concentration areas. All right, so there's a picture there about diffusion, but let's keep going for a second, and there should be some more slides that go through specific definitions for these processes. Okay, So the speed of all of this, like we learned about in the last slide where we talked about the collision between molecules, is impacted by concentration. The greater the gradient between the two areas, the quicker the rate of diffusion. I even think about people that are in a really crowded building when they're trying to go through like two doors. They will kind of spill out from that place, right? So I even think about it with crowds. But the greater the concentration, the faster that movement, that diffusion happens. Also, smaller molecules will move faster and diffuse more quickly. And at higher temperatures, the rate of diffusion is faster. If you've maybe done this at home or want to do it at home, you could take a small beaker or glass of water and compare kind of a cooler glass that maybe you put in the refrigerator for a while versus really hot water from the tap, and then put a drop of food coloring in each of them. The hot one, the warmer temperature, should exhibit faster diffusion. Now, eventually, all the food coloring or whatever your solute is will reach equilibrium, and that's when now there's no more net movement of molecules. There's just this equal kind of distribution of the solute. All right. So I think before we go further here, actually, I'm just looking at how many more slides we've got. I'm going to leave off there and then come back and start back talking about passive membrane transport and go through the specific examples. So we'll kind of leave off here, pick back up with chapter 3 at passive transport.